Welcome back to the University of Melbourne Biology series. This is episode 5, and today we follow the energy. Every living cell is busy. It pumps, it builds, it moves, it copies itself, and not one of those actions is free. They all cost energy, and that energy has to be captured, stored, and spent at the right moment. So this episode is really about two things working together. First, the rules of energy itself, how it flows and transforms inside a cell. And second, the protein machines, the enzymes, that decide which reactions actually happen and how fast. Over the next 12 minutes we will start with raw energy, meet the molecule that carries it, then turn to the enzymes that put it to work in the ways a cell keeps them under control. Let us begin. Let us start with the field itself. Bioenergetics is the study of energy flow and transformation within living organisms. And energy, at its core, is the capacity to cause change, a capacity that can be converted from one form to another. Biological systems run on two primary types of energy. The first is potential energy, which is stored energy, often held in chemical bonds or in a concentration gradient. The second is kinetic energy, the energy of motion, associated with the movement of molecules. The whole story of a cell is the story of shuffling between these two. Energy stored in the bonds of a sugar is potential energy waiting to be released, and when the cell unlocks it, that energy drives the motion and the work that keeps the cell alive. Energy transfer like this is fundamental to every cellular process and to metabolism. Before a cell can spend energy, it has to obey two unbreakable rules, the laws of thermodynamics. Thermodynamics is the study of energy transformations, and two laws matter here. The first law states that energy cannot be created or destroyed, it can only be transformed from one form into another. So a cell never makes energy from nothing. It can only take energy that already exists and convert it into a more useful form. The second law states that the entropy, or disorder, of the universe always increases. Every transformation spills some energy into disorder. This sounds like bad news for life, which is exquisitely ordered, but a cell does not break the rule. It pays for its internal order by increasing the disorder of its surroundings. These two laws are the backdrop against which every reaction we discuss today must play out. Those laws sort every reaction into two kinds. Exergonic reactions release energy and are spontaneous, they run downhill on their own. Endergonic reactions are the opposite, they absorb energy and are non-spontaneous, so they will not happen by themselves. Here is the central trick of metabolism. Exergonic reactions provide the energy needed to drive endergonic reactions. The energy released by a downhill reaction is captured and used to push an uphill reaction forward. One important family of energy-releasing reactions deserves a special mention, redox reactions. These involve the transfer of electrons from one molecule to another, and that electron transfer is essential for producing the cell's energy currency, ATP. So the cell is constantly pairing reactions that give with reactions that take, and electron transfers sit right at the heart of how it harvests energy. If endergonic reactions need a constant supply of energy, something has to carry that energy to where it is needed. That something is ATP, adenosine triphosphate, the primary energy currency of the cell. Look at its structure. ATP is a nucleotide built from three parts, an adenine base, a ribose sugar, and a chain of three phosphate groups. Those three phosphates are the key. When ATP is hydrolyzed, energy is released, and that energy is used to drive the endergonic reactions of the cell. In other words, ATP is the cell's rechargeable battery. Exergonic processes load energy into it, and when the cell needs to do work, it spends ATP to power reactions that could never run on their own. ATP sits at the very center of bioenergetics, the molecule that links energy supply to energy demand. We now have energy and a currency to carry it. But energy alone does not decide which reactions happen. That job belongs to enzymes. Enzymes are biological catalysts that speed up chemical reactions in cells, and they are essential for both metabolism and digestion. Crucially, enzymes do more than make reactions faster. They control them. By determining which reactions proceed and how quickly, enzymes let the cell regulate complex metabolic pathways in a precise and efficient way. Think of the thousands of reactions a cell could run. Without control, that would be chaos. Enzymes impose order, switching reactions on and off and tuning their speed so that metabolism unfolds as a coordinated whole rather than a free-for-all. Speed and control together, that is what makes enzymes the managers of the cell's chemistry. So how does an enzyme actually speed up a reaction? Every reaction faces an energy barrier called the activation energy, the energy hump that reactants must get over before they can become products. An enzyme works by lowering that activation energy. It does this by binding the substrate, the reacting molecule, at a special pocket called the active site. Biologists describe this binding with two models. The older lock and key model pictures a rigid enzyme whose active site exactly matches the substrate, like a key fitting a lock. The more accurate induced fit model describes something more dynamic. As the substrate binds, the enzyme adjusts its shape to wrap around it, forming a snug enzyme-substrate complex. Either way, the active site holds the substrate just so, lowering the barrier and letting the reaction proceed far faster than it ever could alone. Let us zoom into that active site, because its details explain everything. Enzymes are proteins, and the active site is a specific pocket shaped by the protein's fold. Lining that pocket are the side chains of particular amino acids, often called R groups. These R groups are the working parts. They bind the substrate and disrupt its stability, reducing the energy needed for the reaction to occur. Here is a subtle but important point. It is tempting to think the shape of the active site is what matters most, the neat fit we just described. But in fact the chemistry of the active site is more important than its shape. It is the chemical character of those amino acid R groups, what they can do to the substrate, that drives catalysis. Shape brings the substrate close, chemistry does the work. If enzymes drive reactions, then to control metabolism the cell also needs ways to slow them down. That is the role of enzyme inhibitors, molecules that bind to enzymes and reduce their activity, and they play a key role in metabolic regulation. There are several types, and we will take them in turn, starting with the most intuitive. A competitive inhibitor competes directly with the substrate for the active site. While the inhibitor is sitting in the active site, the substrate cannot bind, so the reaction slows. But this kind of inhibition has a weakness, and it is a useful one. Because the inhibitor and the substrate are fighting for the same spot, competitive inhibition is reversible. Simply increasing the substrate concentration lets the substrate outcompete the inhibitor and win back the active site. Flood the enzyme with substrate, and the brakes come off. Not every inhibitor fights for the active site. A non-competitive inhibitor binds to a site on the enzyme other than the active site. That binding causes a conformational change, a shift in the enzyme's shape, which reduces its activity. And because the inhibitor is not in the active site at all, adding more substrate does not reverse it. Flooding with substrate simply cannot displace an inhibitor that is bound somewhere else. The third type is more drastic still. An irreversible inhibitor forms a covalent bond with the enzyme, permanently inactivating it. This is not a temporary slowdown but a permanent shutdown, and it is exactly how many toxins and certain antibiotics work, locking onto an enzyme for good. So inhibition spans a spectrum, from the gentle and reversible competitive block, through the shape-changing non-competitive kind, to the permanent grip of irreversible inhibition. That idea of binding away from the active site to change an enzyme's shape is so powerful that the cell uses it as a deliberate control system. This is allosteric regulation. In allosteric regulation, a regulatory molecule binds to a site on the enzyme that is different from the active site. This binding causes a conformational change that affects enzyme activity. The beauty of it is that it works both ways. Depending on the regulator, an allosteric enzyme can be either activated or inhibited. One molecule binding at the allosteric site might switch the enzyme on, another might switch it off. This makes allosteric enzymes ideal control points. By responding to signals at a site separate from where they do their chemistry, they let the cell turn whole metabolic pathways up or down, which is essential for controlling those pathways and maintaining cellular homeostasis. Allosteric control reaches its most elegant form in feedback inhibition. Imagine a metabolic pathway, a chain of reactions that builds a final end product. In feedback inhibition, that end product inhibits an enzyme early in the pathway. As the product accumulates, it reaches back and shuts down its own production. This is a form of negative regulation that controls the flow of metabolism, and its purpose is clear: it helps maintain homeostasis by preventing the overproduction of metabolites. The cell makes exactly what it needs and no more. And notice how everything comes together here. Energy flows downhill and is captured in ATP, enzymes lower barriers to spend it, inhibitors and allosteric regulators tune those enzymes, and feedback inhibition closes the loop. Energy and control, working as one. That is the logic of the living cell. See you next time.